DNA is the blueprint of life. It contains the instructions needed for an organism to develop, survive, and reproduce. The 4 types of nitrogenous bases in DNA are adenine, thymine, cytosine, and guanine. Each nucleotide consists of 3 parts: a phosphate group, a sugar molecule, and a nitrogenous base. These bases pair in a specific way. Adenine always pairs with thymine, and cytosine always pairs with guanine. These pairs are held together by hydrogen bonds. The backbone of the DNA strand is formed by sugar and phosphate groups linked together by phosphodiester bonds. Phosphodiester bonds connect the 3-carbon atom of one sugar molecule to the 5-carbon atom of the next sugar molecule, creating a strong and stable backbone for the DNA molecule. The process of making proteins begins with the transcription, where DNA is used to make messenger RNA or mRNA. RNA polymerase reads the DNA sequence and creates a complementary mRNA strand. This mRNA will carry the genetic information from the DNA in the nucleus to the ribosomes in the cytoplasm. The processed mRNA now moves to the ribosome, the cell's protein factory, for translation. Transfer RNA, or tRNA, brings the corresponding amino acids to the ribosome based on the codon sequence. RNA polymerase reads the DNA sequence and creates a complementary mRNA strand. This mRNA will carry the genetic information from the DNA in the nucleus to the ribosomes in the cytoplasm. The viral infection cycle begins when a virus attaches to the host cell and injects its genetic material. The viral genetic material takes over the host cell's machinery, forcing it to produce new viral particles. Eventually, the host cell bursts, releasing new viruses to infect other cells.
We have this dude. His name's just going to be Billy for this. And he doesn't want to get polio. So we give him a silly little vaccine, which was ARM. But these work in mysterious ways. This is the polio virus right here. You don't want this. So what we do to make sure you don't get it is we basically get a bunch of these little viruses right here. We put them in a blender type thing, not an actual blender, but I'm just using this for easy mistakes. And they shake. Then you get these little things, the proteins on them called antigens. What we do, these antigens, we basically just pour a bunch of them into a syringe, the liquid into the syringe, so we can make a vaccine. The antigens and other dead debris of viruses go into his arm, ouch, and then into his bloodstream. As you can see here, there's flowing around. Then a white blood cell comes along, or multiple, and they attach here. They'll attach all around it and try to, and then the white blood cell attacks the antigens because they think it's a virus. You're basically getting your body pre-prepped in case you get the virus that just knows what to do. In case the virus ever does happen to like go into your body, this is what will happen. The antibodies that are made by, white blood cells and the attack on the antigens will attack the cell, the virus, it's not a cell, and then kill it, allowing your body, it not to be able to get into your cells and then kill your cells. So that's why we use vaccines, and that's how they work.